Can you guys hear me? All right,
Yeah.
cool. How's everybody doing today?
A little tired.
Why is that?
I'm back to working and my body isn't used to it because I was having so walking 9 hours a day just kind of is knocking me out.
What are you doing working wise?
I just work in a Menards in the paint department, but I walk the length of the store like every.
Gotcha. All right, I need to erase my board because that's from last week's stuff. All right. Alyssa had a question that she sent me via email, and since I was about to talk to you guys when I saw it, I figured I would just address it here because I suspect all of you have the same question. So you guys are working on the on reading. Hopefully, you're working on reading through the GMO report, and hopefully you started on that. And she asked when the written report was going to be done, was going to be due, and I don't have a due date for that yet. I would like it to come in before Thanksgiving, but the actual date is pretty flexible because as long as it comes in before Thanksgiving, I'll have time to read them and thoroughly and grade them and get them back to you before the end of the semester. So it doesn't really matter to me. I think what we should do, and I talked about this a little bit last week when we started talking about genetically engineered organisms, is kind of see where we are in terms of the oral presentations. So if you go and look on, let me share my screen. If you go and look on the Moodle site, which I could have sworn I just had up here a few minutes ago, but I'll go get it. Can you guys see my web browser?
Yeah.
All right. So if you come down here and look at the GMO report assignment, which is right here, you can see that I have now put you guys into groups. One group to handle chapter 4 on agronomic and environmental effects, Branson, McKenzie, Marcus, and Blake, health effects, Jackson, Olivia, Alyssa, and Blair, and socioeconomic effects, Mary, Kenton, and Winston. Those are randomly assigned as I could manage. I just went down the roster and assigned people to 3 different groups. So those are the groups that you'll be working with. Right now on the schedule, the date for those things to be due, for the oral presentations to be due, is next Monday. I think that's maybe a little too soon, and so I think we should actually push that off and then just move the rest of the schedule up a day. But I need feedback from you guys as to when you think you're going to be ready for that. And so I don't want to create an environment where not having a set date just allows you to put it off and put it off and put it off. But at the same time, I also don't want to rush you into doing something before you're ready. So I really need clear communication from you as to what you guys think a reasonable amount of time will be to read through your section of the National Academy of Sciences report, get together with your people and get together a presentation. Last year was the first time that I did this, and I thought it would be a good idea as to how to get people more ingrained into one of the chapters, and also to help the other students to understand the chapter that you were kind of ingrained in. What happened last year was the first day of presentations, they were really terrible. They were a lot of presentations that had like literally hundreds of words on a slide because they just listed direct quotes out of the National Academy of Sciences report and plunked them into a PowerPoint and then proceeded to basically just present us with the words that were in the report. Which there's nothing wrong with the words that are in the report, but if the report was super easy to understand, I would just assign all of it to you, have you read it. We wouldn't talk about it at all in class because I would assume that everybody understood what they read. So the goal of the presentations is for you to take the information that is in the report, distill it down to kind of what the major findings were of that particular chapter, and then present them to your classmates in language that they can understand. If we pretended that everybody could understand everything about the way the National Academy of Science writes about things, then there really wouldn't be any point in all of this. In addition, the thing that you're going to be asked to do is to explain this to your curmudgeonly aunt or uncle, which once again, if all you do is give them the language of the National Academy of Science reports, that's probably not going to be very understandable to them or convincing to them about whatever point it is that you're trying to make. So with all presentations, generally speaking, fewer words is better than more. If you can find ways to make your points visual rather than just with text, that's always better. And then the other thing is with the National Academy of Science report, the chapters are fairly long and they're fairly detailed. You need to take all of that complexity and kind of boil it down into a set of talking points. So think of it as you're giving an executive summary to your curmudgeonly aunt or uncle who knows absolutely nothing about science. And that's a challenge. It's a challenge for scientists to talk to non-scientists about science. I experience this virtually every time I take a bike ride with the group of bike riders that I ride with. So it should be, you should expect it to be a challenge to you as well as non-scientists trying to talk to non-scientists about scientific things. So once again, I want to give you an adequate amount of time to work on the group presentations, but I don't want to put it off without a due date such that you never get started on it. Simply because I know the nature of humans. The nature of humans is to put things off until they actually absolutely are staring you in the face. That said, what seems like a reasonable timeline to you guys? And silence reigned.
I mean, if next Monday seems too soon, you could just extend it to the end of the week, correct?
I could extend it to the end of the week or to the following Monday. Let me pull up the schedule again. So it was going to be. Oh, it was going to be. Oh, it was going to be this Friday. That's way too soon. So how about we put it off until Monday well, why don't we put it off until the 14th of October, the day before the exam? Then what we'll do is we'll just move all of this other stuff up one day. So on Friday, we'll talk about the rise of corn. On Monday, we'll start talking about nutrient cycling. We can get the science out of the way, and then we can just have the day before the test spent on the GMO report. And you should probably plan for about a 10-minute presentation with 5 minutes or so for questions and discussion. That'll take us 45 minutes to get through. And if we spend a little more time in discussion, that'll probably kill that hour. And then that'll be the last thing that we do before we sit down to take an exam on the 16th. that sound reasonable? All right, then the 14th of October it is. I'll revamp the schedule and post another update on the Moodle site. The current schedule has us talking about the evolutionary history of domesticated animals and plants over the next couple of days, and then we'll start talking about the rise of corn, and the discussion of the evolutionary history of plants will actually tie right into the rise of corn. These are two topics that we've had in past semesters that we, for whatever reason, didn't get to, and for whatever reason, we moved through the genetic modification stuff a little quicker than I have in the past. For whatever reason, I'm not sure why. Okay, so that squares that away then. All right. Did you guys go and view the CRISPR video over the weekend? And did you listen to the CRISPR radio stories by NPR about the woman with sickle cell anemia? If so, what questions do you have? What comments do you have? Nothing? All right, I want to go through hold on. I pulled up the CRISPR thing and I figured out over the weekend how to do this, I think. So I just want to go through the CRISPR video with you to make sure that you're getting the things out of it that I want you to be getting. And hopefully I can get the sound to work. Okay. Can you guys see YouTube? Okay. Can you still see YouTube? Okay. Here we go. Can you hear it? Okay. All right. So this is what I want.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA.
So when she talks about letters of DNA, what is she talking about?
Is that like what the strands are connected to?
Okay, clarify. Go into that a little more. What's that?
Isn't it like T and G or something?
Yep. So the letters that she's talking about are these nitrogenous bases, and the nitrogenous bases are the information content of the molecule. So the nitrogenous bases are adenine, thymine, cytosine, and guanine, and these are the things that complementary base pair with one another. So guanine always matches up with cytosine, and adenine always bonds to thymine, and these are connected to a 5-ring sugar, which are then connected to a phosphate molecule. And this phosphate sugar backbone is the side strands, and then the 2 strands of DNA are connected to one another by these nitrogenous bases. So this is a video that is intended for the general public, and so they refer to the letters of DNA, but the letters of DNA are actually these nitrogenous bases. So unfortunate terminology, but I just want to make sure you're translating appropriately.
DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule.
What's this? What do we call the simple pairing rule in biology? This is the pairing rule. What do we call it?
Complementary base pairing.
Yep, complementary base pairing. This rule by which the chemical affinity for adenine to thymine is much greater than the chemical affinity for adenine and guanine or adenine and cytosine, and so they tend to bond with one another. And this is the way by which DNA molecules stay together, and it's also the way by which you build RNA molecules, because RNA nucleotides also have nitrogenous bases. The complementary base pair with the DNA nucleotides of DNA as messenger RNA is being transcribed. So that it's an important concept that they kind of blow through here in 2 seconds. But once again, good review from the stuff that we covered last exam.
A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified.
So this is an interesting graphic because it's basically actually showing you how the nitrogenous bases actually get color-coded in an actual sequencer. So that's actually pretty accurate. And once again, this is the genome. These are the 23 chromosome types that we have, all 23. And then this particular chromosome here is the X chromosome of the sex chromosomes. The Y chromosome is much shorter than this.
Thousands of genes that affect our risk of.
And these colored bars just basically represent the positions of genes on these chromosomes for genes that we know what their function is. And there are tons of the human genome that we know what the function is, and there's also quite a lot of it that we still don't know what its function is, or if it has any function at all. There are certain parts of the genome that we suspect won't ever have any attached function to it. It's just used to be referred to as junk DNA, but a lot of what we used to think of as junk DNA actually has functions that we just didn't realize at the time.
Disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our.
So that big blob is the Cas9 protein. So proteins are oftentimes shown in their three-dimensional structure. Proteins are long chains of amino acids, and then those amino acids have charges on them many times, which allow one part of the amino acid chain to snake around and do intermolecular bonds with other parts of the chain, which then coils it up into these things that we call globular proteins, these proteins with complex three-dimensional structure. And of course, Cas9 is like any other protein. It will have this complex structure. But these are Cas9 proteins coming into the picture here.
Ability to edit the DNA of any species, including humans.
And in this case, you can see shadowed within the Cas9 protein is actually the CRISPR molecule with the guide RNA. If I back it up just a little bit,
to dramatically
you can see the guide RNA is right here that's going to match up with the DNA sequence of this DNA molecule.
Improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA.
So remember that all words that end in ASE in biology or biochemistry are enzymes. And so Cas9 is just an enzyme, and the enzymatic function is that it makes cuts in DNA. So a nuclease is what she just said. Nucleases, they cut DNA. They could also cut RNA for that matter. They cut nucleic acids.
When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA.
So the way that it found that segment of the DNA is once again through complementary base pairing. So I introduced that idea when we were talking about transcription and translation. But once again, it becomes important here in telling this molecule where exactly in the genome of the virus it needs to go.
Labeling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another.
And replacing a mutant gene with a healthy copy is exactly what they did with this woman with sickle cell anemia. They basically replaced one nucleotide in her hemoglobin gene.
Piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, so I talked you through sections of that. Is that video more clear or less clear now than it was before? Unmute yourselves. Everybody unmute. Talk to me.
It's more clear.
Okay, what was unclear before?
I didn't kind of understand how the RNA attached.
Okay, so that's once again just through this complementary base pairing. It's complementary base pairing is a really super convenient because it saves the cell some time and energy in that all it has to do in many cases is make half of it. Well, all it really has to do is make half of a DNA molecule, and the other half of the DNA molecule oftentimes arranges itself pretty well. Same thing with generating messenger RNA. It's just a very efficient way of building ordered sequences of nucleotides in a way that keeps it from making too many errors. What else was clear and unclear about the video for you? Anybody else? All right then. I will just assume that you guys have CRISPR down. Everybody unmute. Tell me what you learned from the NPR article about the woman with sickle cell anemia. Kenton. What did you learn about the sickle cell anemia case?
Sickle cell anemia primarily occurs in people of African descent in the United States.
Sure. Yep. One of the things that sickle cell anemia does is it imparts some limited immunity to malaria. And so you find the sickle cell gene to be more common in people from areas that are prone to high incidences of malaria. And so it's one of these genes that the hemoglobin gene is one of these genes where one form of the gene works well for people under one set of circumstances, but then it has these other negative effects when you're not being affected by malaria, for example. What else did you guys learn? Mackenzie, talk to me.
Well, I didn't even know what sickle cell anemia was, so just learning that like red blood cells kind of like disappear was interesting to me.
Okay. Anyone else?
I thought it was interesting. They said that like they had to do chemotherapy on her to make room for the cells. I didn't know that they had to do that.
Yeah, so when you're making blood cells, they get made in your bone marrow. And so one of the things that they had to do was they had to get rid of all of the stem cells that create the defective hemoglobin. And so they essentially had to do chemotherapy to kill her cells that would normally make blood, which would usually be a really bad thing to do to somebody. But what they did was they took some of those stem cells, they took them out of her, they CRISPR'd them to create healthy hemoglobin, and then they put those stem cells back into her. So it was pretty risky going into an experimental treatment where they basically destroyed all of your, what we call the hemopoietic tissue, all of the tissue that makes your blood supply, and basically killed it off with the promise that we're going to put it all back, and what we put back is going to be better than what was there before. So yeah, it's a pretty crazy thing to do to someone. Yeah. What else? So if they hadn't done that, what would have happened? If they hadn't killed off our old cells that create blood cells and just put new stem cells in there that would create new blood cells, what would the net effect have been?
Would they have not been able to integrate into her system as efficiently?
Well, it isn't that they wouldn't have been able to integrate. They would have been able to integrate just fine because they're her cells. But what would happen with her blood?
Become overcrowded?
So you would have tons of these cells that were producing the sickle cell anemia style hemoglobin, and relatively few of these new cells that create the corrected hemoglobin molecule. And so the CRISPR'd cells would just be swamped out by the abundance of the other cells in terms of the total number of blood cells that were produced. So they had to do that in order for the CRISPR'd stem cells to take hold and produce enough blood for her to actually have enough of the corrected hemoglobin that it would actually alleviate the disease in this case. What other thoughts do you have about the 2 radio stories? In the second story, what were they basically doing?
They just checked back up on her a year later, pretty much.
Okay, and how is she seeming to do under all of this?
She said that like pretty much all of her pain was gone, so it seemed like it worked.
Yeah, so it was a pretty risky procedure, and it seemed to have worked, which is actually kind of amazing that something would work the first time you try it in a human. Now, one of the problems with this is that no guarantee that it will work the second time. So we have a sample size of 1, and you never want to go into something with just a sample size of 1. But it's one of these things where this particular technique has now, shown some promise at alleviating a genetic condition in a person who's already undergone all of their development. So you can actually go in after the fact and correct a person's DNA to alleviate a particular malady that is strictly genetically determined. It's a genetically caused malady. So in that way, it's a pretty interesting and newsworthy sort of experiment. Would any of you guys do that if you had sickle cell anemia? Would you go for it?
No, I would not.
Oh, why not, Winston?
I would just not want to be a guinea pig, I guess, because I mean, I'd like there to be some further research before I risk that for myself.
Yeah. Okay. So, and that's actually smart. You guys, I'm sure, have been following at least a little bit of the news about COVID. What's one of the concerns? So just to relate this back to something that's going on in America today, what's one of the concerns about vaccine development for COVID vaccines?
Notice the vaccines, and it hasn't been like you could have a negative reaction.
What was that, Mackenzie? I couldn't quite hear that.
That was actually my roommate.
Oh. What does your roommate have to say about it?
He said they're manufacturing like a whole bunch of vaccines. What else did he say?
And like people could have a negative reaction to them.
And people could have a negative reaction to them.
Yeah, and so one of the things that you have to do with a COVID vaccine is you have to test it out on people. And some of these pharmaceutical companies are having trouble finding what?
People to test it out on.
What's that, Blair?
Willing volunteers.
Yeah, they're having trouble finding volunteers because nobody wants to be the first person to take the vaccine. And so, Winston, I respect your decision to not go for the sickle cell anemia CRISPR treatment. But there does come a point where some brave souls have to step up and say, okay, it's been through phase 1 trials. Now we're into phase 2 and 3 trials, which are expanded trials based on giving it to humans to see if it works. There is going to have to be somebody who volunteers for those things. And if everybody just refuses to volunteer because they're afraid of what the vaccine may do to them, then we're not going to make much progress, unfortunately. So there are certain companies that are trying to get like phase 3 trials. So the process for getting any treatment approved is you go through a set of trials, usually in animal models like mice and things like this, to see if it's safe, to see if it's toxic, to see if it's going to kill the individual. Then you go through a set of trials that looks for side effects that aren't fatal or dangerous, but just it would be nice to know what the side effects are so that you can look out for interactions with other conditions people might have. And then there are a set of trials at the end that says, okay, it's safe, there aren't too many side effects. Now, does it actually work? Does it actually do the thing that it is designed to do? In the case of CRISPR, did it actually cure this woman of sickle cell anemia? That's the efficacy part of it. In the case of the COVID vaccine, does it decrease your chances of contracting COVID? So when they go in and do this phase 3 trial, the efficacy trial, they have to have enough volunteers, and the volunteers have to be the right kinds of volunteers. People who are not particularly good prospects for the right type of volunteer are college professors, because I made the conscious decision to not interact with any of you. I said no to that at the beginning. I said, I'm not going to do that, and I'm not going to have you guys interacting with one another. When it came down to the math of whether we meet in person or online, I said, there's going to be a 100% chance of inconvenience. And I had to balance that against a non-zero possibility that one of you would get sick and maybe even die. And when I thought about it that way, the math became really easy for me. And so I went to online and said, we're just going to work through the inconvenience of it. But because I'm isolated from you and from other people, everybody except the person that I live with, essentially, I'm not a very good candidate, because giving me the vaccine won't show whether or not it's effective, because I probably am not coming into contact with people who have COVID. Public school teachers who have to teach classes with students in their classroom, 8 hours a day, 5 days a week, those people would be excellent candidates for a vaccine trial, because the chances of them coming into contact with other people who have COVID are relatively high. And then you could look at what is the incidence of getting COVID in people who have the vaccine versus people who don't have the vaccine. And so you not only need enough volunteers, you need enough of the right kind of volunteers, those that are in high-risk occupations that are likely to come into contact with other people who carry the disease. So it's tricky, all of this health development stuff. And in the case of the CRISPR thing, they have one person that they have tried this with, and only one person as far as I know. Questions about the CRISPR'd sickle cell anemia patient? Mackenzie, thank your roommate for her input.
I will.
Okay, good. All right. Let's move on then from CRISPR. So as a kind of a companion to talking about genetics and genetic engineering and genetic modification, we have a set of crops, plant and animal crops, that we grow on farms and ranches and things like this in order to feed the human populace. And so it's useful sometimes to get a handle on where did these things come from. So this is a section of the class that verges once again into an area of biology that you're maybe not familiar with, but evolutionary biology. And it deals with how we know how organisms evolved in the past. So oftentimes you'll hear people talk about the evolutionary history of organisms, and you might wonder, well, how can scientists possibly know what the evolutionary history of a group of organisms is, because you don't have a time machine. You can't go back in time and see what organisms were like, 10,000 years ago, 100,000 years ago, 1 million years ago. But as it turns out, biologists have developed a set of techniques that allows them to do this. And so I want to expose you to those techniques so that there's nothing mystifying about them. And then after I've exposed you to the technique, I want to then talk about today the evolutionary history of animals, that we use for crops, and then on Wednesday we'll talk about the plant side of things. And when we talk about the plant side of things, we'll also talk about some ideas from a guy named Jared Diamond, who put out some ideas about the effect of the evolutionary history of crop species on human history. And so hopefully that will also be engaging for you. You guys can see my PowerPoint? Okay, cool. So learning goals for the next couple of days are, I want you to understand how biologists infer evolutionary relationships among organisms. And you can do this using morphological characteristics or behavioral characteristics or genetic characteristics. It doesn't really matter. The same method works regardless of what the source of your data are. I also want you to understand the origins of our major food crops, both plant and animal. So our food crops came from geographically discrete areas. So for example, tomatoes came from the New World. Pigs are an Old World organism. Potatoes come from the Andes. So there are places where these crops grew originally out in nature, and then when humans found them useful, they got moved around a lot. So tomatoes and squash were found in Mesoamerica, southern Mexico, Central American area, and corn spread throughout the world over time because it just turned out to be a super useful crop. Developing an understanding that the food that we eat is the result of the evolutionary history of our relationship with plants and animals, and it's due in part to this biogeographic happenstance. You know, the people of the Andes had potatoes because those potatoes happened to already be growing there when people arrived. People in Ireland didn't become reliant upon potatoes until after people in Europe had discovered the New World and then brought those crops back to the Old World. So when you think about Italian food, somebody described to me their favorite Italian food dish. Jackson, what's your favorite Italian food?
Probably fettuccine alfredo.
Fettuccine Alfredo. Well, that's not any good because there's no tomatoes in that. Somebody else give me a tomato-based dish that they like.
Lasagna.
Okay, Winston, you like lasagna. So lasagna has red sauce. In red sauce, whether there's meat in there or not, red sauce is primarily made up of tomatoes. So when we think of Italian food, we oftentimes think of spaghetti and ravioli and things like this that are drenched in red sauce. But because tomatoes are indigenous to Mesoamerica, southern Mexico, Central America, the Italians didn't have red sauce prior to 1492 because tomatoes were not available to people in Italy until after the New World was once again discovered by Europeans. And so our kind of stereotypical view of Italian food is a relatively recent sort of development in Italian cuisine, simply because they didn't have access to tomatoes before a certain point in history. So the history of things becomes important. And then at the end of class on Wednesday, we'll talk about Jared Diamond's idea about the reasons that certain peoples came to dominate the world. And his ideas basically grow out of this biogeographic happenstance. Which people lived in which parts of the world that had a suite of plant and animal crops that allowed them to grow in population size, found city-states, develop governmental structures, et cetera, that then allowed them to go out and kind of conquer the world. And so he has a very ecological, evolutionary, and food-based set of ideas about why certain cultures came to dominate global commerce and government and things like that, and other areas of the world did not. So we'll talk about those at the end of class on Wednesday. So I'm sure all of you have seen an evolutionary tree like this at some point. Well, I don't know, maybe you haven't. Have any of you guys ever seen an evolutionary tree like this showing you how organisms are related to one another? No, never? Okay. Well, this is a common way of depicting this. And so we have the ray fin fishes that are more closely related to the lungfishes. Salamanders and frogs are more closely related to one another than they are to anybody else. The reptiles, turtles, snakes, lizards, and crocodiles are all here together. The birds are here, and then we have the mammals out here at the end. And so one question that you might have when you see an evolutionary tree like this is, well, how do biologists know that this is the way that these major groups of vertebrates, in this case, came to be? And the way we do this is we use current morphology to extrapolate to the past. And I'm going to show you how we do that. So this whole thing is called phylogenetic systematics, is the area of science that is involved. And what we're trying to do is we're trying to create what's known as a phylogeny, which is just this tree-shaped structure that shows the evolutionary relationships among organisms. And so what we do is we start off by creating what's called a character matrix. We basically get all of the organisms that we're interested in recreating their evolutionary history. So we have a lamprey, which is a jawless fish. We have perch, which is a bony fish. Salamander, which is a type of amphibian. Lizard, which is a type of reptile. A crocodile, which is also a type of reptile. Pigeons, and a mouse. Pigeon is a bird, and a mouse is a mammal. And then we say, okay, well, we have a bunch of, in this case, morphological characters, and we can ask ourselves, does a lamprey have jaws? It doesn't. Does a perch have jaws? Yes, it does. And everybody else has jaws. Who has lungs? Well, lampreys and perch both have gills, and salamanders and everybody else have lungs. Who has claws and nails? Well, lizards, crocs, pigeons, and mice do, but lampreys, perch, and salamanders do not. Who has a gizzard? Well, only crocodiles and pigeons. Who has keratinous scales? Well, lizards, crocodiles, and pigeons. And so we create this matrix of morphological characters, and we ask ourselves, which of these characters has been evolutionarily derived? And in order to do that, we have to decide who's the outgroup, who's the earliest group of organisms in terms of their evolutionary history of when they arose. And in this case, the lamprey is the outgroup. So we use the characteristics in the lamprey to what we call polarize each of these characteristics. So the lamprey doesn't have any of these characters. So that's the ancestral condition. And when you gain a character, then that character presumably arose as a result of evolution. And so we polarize our characters and we classify them as either ancestral characters or derived characters. And we ask ourselves, well, which of these derived characters group large groups of the taxa that we're trying to look at? So when we first look at this table, obviously jaws are really important at grouping because jaws separate lampreys from everybody else. So in the first iteration, what we would do is we would say the lamprey is the outgroup, and what separates the lamprey from all of the other groups we're interested in is the presence of jaws. Lamprey doesn't have them, and everybody else does have them. Then we can go back to the matrix and say, well, what's the next biggest character in terms of the number of groups that it includes? What would that be? Not a rhetorical question.
Whether or not they have lungs.
Whether or not they have lungs. In this case, lungs are not found in lamprey and perch, and they're found in everybody else. So we would say that perch was the next group that evolved, and what separates perch from all the other groups is the presence of lungs. The perch and everybody else all have jaws, but what distinguishes all the other groups from perch is the presence of lungs. So now we've accounted for 2 of these characters. What's the next most useful character for grouping large numbers of the remaining taxa?
Claws or nails.
Claws or nails. Who was that, Blair? Good. All right, so now we can say, well, salamanders don't have claws or nails, but everybody else does. And so salamanders evolved after perch because they have lungs, and they evolved, and the perch evolved after the lamprey because perch have jaws. But then we still have all of these other groups to concern ourselves with. So we go back to the character matrix. What's the next most informative character that we can look at?
Keratinous scales.
Okay, so scales, these epidermal scales that snakes, crocodiles, and birds have. You don't think of birds as usually having scales, but they do still have scales on their feet. And so when we do that, that means that the mouse doesn't have that characteristic. And so the mouse would come out, and it would leave everybody else with keratinous scales. So we have the mouse evolving before all these other groups, salamanders evolving before mice, et cetera, et cetera. Now we've taken care of those. So now what is the next most informative character? Is it the gizzard? Is it feathers? Is it hair? Is it mammary glands? In terms of grouping multiple taxa.
It's the gizzard.
Okay, so it's the gizzard, and the gizzard is found only in crocodiles and pigeons, which means that now lizards are left out of that grouping. So now we have lizards left out of the grouping, gizzards appeared, and gizzards is what separates the pigeon and the crocodile from all of these other groups. Now, is there something that separates pigeons and crocodiles?
Feathers.
Yeah, the presence of feathers. And so we would put feathers down here, and that's a characteristic that separates crocodiles from pigeons. And so this is the evolutionary history of some of the vertebrate groups, a good number of the vertebrate groups. Jawless fishes, jawed fishes, amphibians, mammals, reptiles, crocodiles, and birds. But does this account for all of the characteristics on the tree?
Nope.
It doesn't, because we have hair and mammary glands that are only found in mice. But they don't serve to separate mammals from the other groups. They're just characters that evolved along this evolutionary history of mammals. And so we would put these on this branch of the mammal branch, because they occur in mammals, but they don't occur in anyone else. And because they don't occur in anyone else, they don't help us see the shape of the tree. They don't help us see the relationships among other things. They're characters that only occur in that one terminal taxon. So there are 2 kinds of characters that we've seen here. One character is a derived character, a character that arose through evolution, but it's shared by a group of taxa. So shared derived characters are useful because they help us with this grouping that we just did. These are called synapomorphies. And those are the things that are useful. So shared derived characters are useful. Autapomorphies these are characters found in only one taxon. And that would be things like feathers in the case of birds, and it would be things like hair and what was the other thing? Hair and mammary glands in mammals. and they're not useful for discerning the shape of the tree. And so this is what we call a parsimony analysis. And the reason that it's called a parsimony analysis is it looks for the simplest explanation to account for all of the data. So one of the things that we can do with this tree is we can go through and count the number of evolutionary transitions that there are here. So there's 1, 2, 3, 4, 5, 6, 7, 8. 8 evolutionary transitions. Actually, in the language of parsimony analysis, there are essentially 8 steps that evolution goes through that describes this tree. And parsimony basically says that trees that have the fewest numbers of steps are the trees that we should trust the most. And you can argue with whether that makes sense, that the simplest explanation is oftentimes the correct one or not. Biologists themselves have been arguing about. Now, a lot of times people like to think that mammals and birds are more closely related to one another than other groups. Why do we think that oftentimes? This is a common misconception that people have. We think that we're similar to birds, largely because of one characteristic that birds and mammals share. Why would you think that mammals might be more closely related to birds than mammals are to salamanders and lizards?
Maybe like hair, like the feathers on them.
Okay, so hair and feathers actually are both modifications of epidermal scales, as it turns out. So this is not the best this is not the best set of characters to use, because keratinous scales evolved into feathers and keratinous scales also evolved into hair. But that's a pretty geeky refinement of the thinking. What else is shared among mammals and birds that we think about probably fairly often? If you've ever held a bird in your hand. Maybe you've never held a bird in your hand. Maybe you ask, you should come to my house and visit my chickens. Anybody ever held a bird? They're all warm-blooded, and we're warm-blooded, and so oftentimes we say, oh, well, that's a physiologically a pretty big deal, being warm-blooded. All the rest of these organisms are what we call cold-blooded, or they're ectothermic. They get their body heat from the environment. We generate our body heat metabolically. And so sometimes people would want to take the mouse and the bird, and they would want to put them next to one another on this evolutionary tree. But in the process of doing that, we would have to have all of these things like keratinous scales and gizzard. They would have to evolve to account for them being in lizards and crocodiles, and then they would have to unevolve and evolve away, basically, for them to not then be present in mammals. And so that would add to the number of steps on the tree. It would make the tree have more evolutionary transitions, because you would have to evolve keratinous scales in a gizzard, and then you would have to evolve them away in the mammal line. And so you would have 2 extra steps to your tree. That tree would now have 10 steps instead of 8 steps, and so we would favor the tree with only 8 evolutionary transitions. As it turns out, to put mammals and birds right next to one another, if you looked at a much broader data set of characters, it would require all sorts of weird compromises like that across the entire tree. And so we generally don't do that, because it's not parsimonious. It's not the parsimonious way of thinking about evolution. So that's how we generate this tree. Now, I'm not going to ask you to take a data set and generate a data set, but I want you to understand where these evolutionary trees come from so that when you see a tree here in a minute, you know what's going on. So when we start talking about the origin of crops and domestic animals, we can think about all kinds of organisms that we have domesticated in the service of agriculture. We have donkeys, camels, various types of camels, Arabian camels and Bactrian camels. We've got cattle, we've got chickens, we've got goats, we've got goose, horses, llamas, pigs, reindeer, et cetera, et cetera. And what you see is that all of these organisms came from different parts of the world. Pigs originated in China, actually, and then migrated into Europe with the retreat of glaciers after the last ice age. Turkeys are indigenous to the New World. Cattle are indigenous to the Fertile Crescent area. Reindeer, of course, are obviously indigenous to far northern areas. And in the same way that you have animal crops that were specific to particular places, you also have plant crops that were specific to particular places. And so we see cacao is a New World crop. We see sweet potatoes were found in, well, where are sweet potatoes on this stupid map? Oh, sweet potatoes are Mesoamerican. We see sugar, sugarcane. Where is sugarcane? Oh, sugarcane is not on here. Sugarcane is actually indigenous to the highlands of Papua New Guinea. Corn and tomatoes and things like this are found in, once again, Mesoamerica. And so every crop came from a particular biogeographic place in the world. And so the question is then, well, how did we get all of these crops into our current mix of food? And we're going to start looking at this by looking at, whoa, something's wrong here. We're going to start by looking at animals. So these are the major hoof-stocked animals that people use for food around the world. And so we have the African buffalo that is used for food and milk, but is also just used in many cases as a draft animal to pull plows. We have bison. There's a North American bison, and there's a thing called a wisent, which is found in Northern Europe. They look very similar to one another because you would expect them to be, and as a result, you would expect them to be closely related to one another evolutionarily. This is mainly, the American bison is an animal of the plains. The wisent is, for the most part, a forest-dwelling organism. We have the yak in the Himalayas. We have water buffalo in Southeast Asia. We have the banteng, which is a hoofed animal from Australia, one of the few non-marsupial mammals in Australia. We have the gar, which is this large buffalo-looking animal from Southeast Asia. And then we have the modern beef cattle that we are familiar with, Bos taurus and Bos indicus. Bos taurus is a European version of the cow, and Bos indicus is an Indian subcontinent version of the cow. But both of these cattle presumably came from this thing called Bos primigenius, which is an ancestral cattle that is found in old caves and is depicted on the walls of caves and cave paintings and things like this. So these are the players. What we'll do at the beginning of class on Wednesday is we'll talk about the evolutionary relationships among these players and where they occur in the world and how that relates to the evolutionary history of why certain people use them for food and other people don't. Questions? All right, then. I'm working on your exams now. I hope to have them back to you on Wednesday. I don't see any reason why I shouldn't have them back to you on Wednesday. And I'll just, when I'm finished with them, I'll actually just email the digital versions of them to you. And that'll be that. And I'll see you on Wednesday.